Good evening, Abhay. Let's wait for 2 minutes, then we will start at 7. Okay, good evening, everyone. So, to start with today's session, first of all, I want to ask you one question. It's regarding today's session. So if I give you one protein sequence, how will you know whether a similar protein exists in another organism? I'm repeating my question. If I give you one protein sequence, how will you know whether a similar protein exists in another organism? Can you answer this question? Anything that comes to your mind? Very good, Dolly. Very good, Dwaipayan. Very good, Sudeshna. Very good, Siddesh. Very good, Anmol. So you are understanding the question, right? What question I asked you? What does the question mean? Yes, very good, Sudarshana, by checking the sequence similarity. So you can answer this question using BLAST by checking the sequence similarity. So let me share my screen. Ashritha, I asked the question that if I give you one protein sequence, how will you know whether this protein sequence exists in another organism? That's the question that I asked. So very good everyone, using BLAST and by checking the sequence similarity. Very good Vivek. So first of all, let us understand what is sequence similarity. So, in biology, similar sequences often mean similar function. It means if two proteins look similar in sequence, if two proteins have similar sequence. So, this means that they may have come from the common ancestor and they perform similar biological roles. Just like we have similar words in different languages, but their meaning and their use is the same. In the same way, sequence similarity means two protein sequences performing the same function because their sequences are similar. Now let us understand what is BLAST. So BLAST stands for Basic Local Alignment Search Tool. Basically, BLAST compares one sequence, which is our query sequence, against millions of sequences in the database. So what does BLAST do? It compares our query sequence against millions of sequences in the database. So BLAST answers one simple question, that where else does the same sequence exist? So BLAST answers this very simple question, where else does the sequence exist? So we have mainly 5 types of BLAST, but as we are working with protein sequences from UniProt that we downloaded yesterday, so today we will use BLAST P. Let us open our VS Code. So we have already downloaded the insulin protein sequence from UniProt yesterday. So today, this entire block is our query sequence. So what is query sequence? The sequence that we already have. So we are going to find out the sequence similarity of our query sequence using BLAST. So let us open Google. Here we will search NCBI. Now here, when you search NCBI, you can directly see here BLAST. Or if you want to go inside the NCBI database, Here you can see the BLAST here. So we are saying that BLAST is the NCBI tool. So we can directly use BLAST here from the NCBI. So let's select it. Now here this is the BLAST page. So as you can see, there are 4 types of BLAST. We have nucleotide BLAST, we have protein BLAST, we have BLAST X, and we have T BLAST N. So the meaning of these 4 types of BLAST are already written here, that is, it is nucleotide BLAST. So it means that the query sequence will be a DNA sequence, means our query sequence will be a nucleotide, which we will be comparing against nucleotide database. So same here, protein BLAST means our query sequence will be a protein sequence, which we will be comparing against protein database. And here we have BLAST X. So here in BLAST X, it converts the DNA sequence into protein sequence and then compare it with protein database. That's why it's written here translated nucleotide, which is compared against protein database. And then we have T BLAST N. So it compares protein sequence against translated DNA sequences. So that's what written here, that the query sequence will be protein sequence and it will be compared against translated nucleotide. So as we have a protein sequence, which is our query sequence, so today we will select this protein BLAST. Let's select it. Now here, either we can type here the accession number of our query sequence. We already know what's the accession number is. Let's open our FASTA file. So this is our accession number. Either we can type this here in this blank area here, or we can paste our sequence as well, or we can choose the file. So let's choose the file. Let's click on the choose file option. Then here we have the file insulinprotein.fasta. Now we have uploaded our file insulinprotein.fasta and this file contains our query sequence. Now here you have to do nothing. Simply you have to click on BLAST button here. So now you are seeing that the BLAST is running. Let's wait for 2 minutes. Now we are seeing the result that BLAST has given us. If we scroll down, this is the result that BLAST has given us. So first of all, let's see. This is our query sequence. This is the sequence ID of our query sequence. It is P01308 INS human insulin, which was our query sequence. Now, what has BLAST given us in the result? So all that you are seeing here, these are protein sequences. These are protein sequences which have matched with our query sequence. Means these are similar sequences with our query sequence. So the sequences which are at the top, so this first sequence is at the top, means it matches 100% with our query sequence. Means this sequence and our query sequence, they match 100%. You can also see here 100%, 100%. Then as you scroll down, as you went down, so here you are seeing 78%, 78%. So as you scroll down, there is a less match. Okay, so if you want to choose a sequence similar to your query sequence, then you will choose from the top. Here we have the accession number of all these sequences as well. And here the main point in this BLAST is E value. You always have to see E value. For now, let's just understand the lower the E value, the stronger the matches. Okay, we are not going deep into it. We are just taking an overview. How does BLAST is being performed in NCBI? Now, we have performed BLAST in NCBI, but as we are working with Biopython, we are doing programming. So all the process that we have done here, what we did, we used BLAST and we found out all the similar sequences to our query sequence. Now all this process we are going to do using Biopython in our Python using BLAST. Is there any doubt till here? Okay, so before we open our VS Code, I want to ask you one question. If human insulin matches the insulin, okay, Vidasri, what do you want me to repeat? Vidasri, what do you want me to repeat? Okay, see, this is the BLAST page. Here is the information. This is our query sequence, and all that data, all this data you are seeing here, these are sequences. So these all sequences are similar to our query sequence. Similar means they have similar amino acids. So all the sequences that are on the top, so they are a very stronger match. As you go down, these matches less with our query sequence. Okay, so here you have the accession number as well, you have the E value as well, but we are not going into deep today. We are going to learn how we can do this in Biopython. Okay, Vidasri? Yes, so before we move forward with our Biopython, I want to ask you a question that if human insulin matches the insulin in mouse and it also matches the insulin in cow, then what does that tell us? What do you mean by that? If the human insulin matches with the insulin in mouse and it also matches with the insulin in cow, then what do you mean by that? Very good, Riddhi. Conserved sequences are present. Anyone else? Very good, Ananya. Very good, Sudeshna. Very good, Atharva. Yes, Vedasri. Very good, Sudarshana. Very good, Dennis. Yes, so this tells us that the insulin is conserved. It is biologically important and it is similar across species. So this shows that insulin is evolutionary conserved, meaning it is a crucial protein that has remained similar across different species over time. So finding the sequence similarity with BLAST, it helps identify important genes and proteins in new organisms. So that's why we are learning BLAST. That's why we are learning about sequence similarity. So let's open our VS Code. So till now we have understood 2 points very well, that BLAST is an NCBI tool. BLAST always needs a sequence as input. That input can be a DNA sequence, it can be a protein sequence, and Biopython will help us use BLAST through Python. So first of all, what we will do? We will import from the Biopython library. So from the Biopython library, we are going to, okay, from Biopython library, inside the BLAST module, we are going to import NCBI www. So here, BLAST is the module inside Biopython library, and inside this BLAST module, we have imported NCBI www module. Now, if you read here, if I hover my cursor over NCBI www, here you can read this. It is written code to invoke the NCBI BLAST server over the internet. This module provides code to work with the www version of BLAST provided by the NCBI. So this means that Biopython does not run BLAST locally. It sends our query sequence to the NCBI servers via internet. Okay. So if you want to perform BLAST using Biopython, you must have a internet connection. Now, next, what we are going to do, we are going to make a variable. So let's make a variable. result underscore handle. So I have made a variable result underscore handle. Now we are going to use this module. So I'm writing NCBI www. Now inside this module, we have a function. We already know how to call a function. We add dot operator. So this means inside this. So inside this module, we have a function qblast, and we add parentheses because this is a function. Now why we have called this function? Because this function is going to send our query sequence to the NCBI blast. Okay, now inside this we will give our input. Now what input we will give? We will give our query sequence. We will give the input of what database we want our query sequence to compare against. Let's open BLAST again one more time. So here when we open the BLAST, we selected the protein BLAST. Okay, so that means we are going to give this argument inside our code. So here I'm going to write that I want the program BLAST P. So here P stands for protein. So I have selected the program BLAST P. Just like we selected here protein BLAST, same way we are giving the argument inside our code. So here I have given the input of the program BLAST P. Now, as I said, our query sequence is a protein sequence, which means we want to compare it against the protein database. So here, next argument I'm going to give about the database. So here, I am writing the database NR. So here, NR means non-reductant protein database. Okay, NR stands for non-reductant protein database. Now, what do you mean by non-reductant? It means because it removes the duplicate sequences. So, NR contains many protein sequences from many organisms. So, it removes the duplicate sequences. So, BLAST runs faster and it gives cleaner result. So, that's why NR is the default and the safest choice for protein BLAST. Next, we will give our query sequence. So here I will write sequence. Now we already have our query sequence here. So let's just copy this sequence and paste it here. So we have given our three arguments. We have given the program, we have given the database, and we have given our query sequence. Now here, we have already read here that what does this module do? It sends our query sequence to the NCBI blast via the servers. So we have written this program. Now what is going to happen? We are sending, by writing this program, when I will click on the run button, we are sending our query sequence to the NCBI blast. And then the NCBI blast will return us the result, and the result will get saved inside this variable. Okay, let's learn one more thing. If I write here print result underscore handle. So what I am doing here, I am printing my variable. So if you are thinking that when I will click on the run button, what it will print, you are clearly seeing that inside this variable, I have stored this program. So if you are thinking that Python will print me this, then you are wrong. We just learned now that what this program is doing. This program is sending our query sequence to the NCBI blast via the servers. So when I will click on the run button here, let's run it. Now you are saying nothing is printing. It does not mean that this has printed nothing. It means that it is under work. The sequence here is getting sent to the NCBI BLAST. The work is being done here in the background. So here it means that the Biopython is working, and as soon as the NCBI will send the result back in this variable, Biopython will print us all the data that is saved inside this variable result underscore handle. So for now, I'm going to kill this terminal because as we see in the BLAST, in the NCBI, the result was too long, right? So now we have seen that the NCBI will return us the result in this variable. So now what we have to do, we can't directly print this variable. We have to open a file and we are going to save the result which NCBI will send us in that file, and then we will read that file. So to open a file, here we will use a keyword with. Now whenever we want to open a file, we will use a keyword with. Now here I am using a function open because I want to open a file. So whenever we want to open a file, we will use the keyword with, and then we will use function open, and then we will give our input. We will give the input of our file name, which file we want to open. So here, let's write the file name. I am making the file name blast result .xml, and I am also giving the format of the file that I want the result in blast result.xml. Okay, now as you can see that this file name does not exist inside my folder here. So what is going to happen if this file name already exists inside my folder? This line of code will open this file, and if this file does not exist, this line of code will create that file automatically and will open the file as well. Okay, so this with option, with this with keyword, this line can create our file as well and it will open our file as well. Now there is one more input that we have to give. We have to open our file in a mode. So we are going to open our file in write mode. So everything that you are seeing here, these are modes, that in what mode we want to open the file. So as we want to save our result in this file, so I am opening this file in write mode. So it's very easy. W is for write. Okay. So what I have done here, I have given the name of my file and I have opened it write mode. Currently, this file is not created because I have not clicked on the run button. Currently, I'm writing the program that I want to create this file and I want to open this file, and then I want to open this file in write mode because whatever result NCBI will send me back in this variable, I want to save it inside this file. Now, here I'm writing as B. Now, what does this mean as B? It means that I am giving my file, this blast result.xml, I am giving my file a nickname. So I want to open this file as B, and then we will add colon and press enter. So this program means we have opened our file. This is our file name, and here I have given my file a nickname B. So by this line we have created our file. We have opened our file. Now what we have to do next? We have to write the program by which all the result in this variable will get saved inside this file, which I am opening as B. I am giving it a nickname. So instead of writing this full name again and again, I will just use B. Now here I will write B because we want to save the data in this file, right? That's why I am calling a function write. So I want to write all the data in this file. So from where I want to write the data in this file? I want to write all the result in this file from this variable because NCBI will send the data here. So inside this parenthesis, I will give the input of my variable. So let's write the name of our variable result underscore handle. Now, this program is still not complete. There is one more thing missing. So, what is missing? Here, we have to read this variable. We have written here that we want to write anything, every result that is present inside this variable, we want to write it inside this file B. But first of all, we have to read this variable. So that's why here we will call the function read and parenthesis. As read is a function, that's why I have added parenthesis. Now this program is complete. Do you have any doubts how to open a file? How I have written this program? Do you have any doubt? Do you want me to repeat? Okay, Parthiv. Okay, Siddhesh. Okay, Dwapin. Okay, let's move to first of all this. So these are the three arguments that we have passed inside here. We have passed this as an input. So first of all, just like I told you that we are using blast P. So when we opened the blast, no, Abhi, it will not replace the older file. I will repeat it. So here, just when we open the blast page, here we selected the protein blast because our query sequence is a protein sequence, right? So what we did here, we selected this. We selected this protein blast, and here what is written? Protein and protein. So this means the query sequence is protein, which is compared against protein database. So we have given these two arguments inside our code. So here we have given the argument that our program is blast P. It is not blast N, it is not blast X, it is blast P. So this is our first argument, and then the second argument that this is a query sequence against which collection of sequences do we want to compare our sequence. So we want to compare this. This is our query sequence. We want to compare our query sequence against protein database. So here I have written the database NR. So NR stands for non-reductant protein database. So non-reductant means it contains the protein sequences from many organisms and it removes the duplicate sequences so that BLAST can run faster and so that it can give us cleaner result. Okay, that's why we have written here NR, which stands for non-reductant protein database. And then we gave the argument of our query sequence. Here we pasted our query sequence. Now just like we have learned here, I wrote here print and the name of our variable result underscore handle. So I showed you by clicking on the run button that this variable does not print anything that is written inside this variable. Python did not print all of this inside my terminal. Python did not print me anything. So we just learned that it doesn't mean that it is not printing anything. It was meant that the work is under, means the work is being going on. So when the NCBI will send the result back, it will get stored inside this variable result underscore handle. This is my variable. So when by clicking on the run button, what happened? NCBI here, by writing this program, we are sending our data to the NCBI. We are sending our sequence to the NCBI server via the internet, and when the NCBI will give us the result back, it will get saved inside this variable and it will get printed inside our terminal. But as I said, the data was too long. We cannot directly print all the BLAST results. So we have to save the data sent by the NCBI in a file. So that's why we have opened a file. So whenever we want to open a file, we will use the keyword with. Okay, whenever we want to open a file, we will use the keyword with, then we will use the function open because we want to open the file, and then we will give our input of the file name. As I have written here, the file name blast underscore result and the file format .xml, that I want the blast result in the XML format file. So as you can see, this file name does not exist here inside my Biopython course folder, right? This file name does not exist here. So what will happen? This program, this line of code will create the file with this name as well, and it will open it in the right mode. Here we have given the mode, that in which mode do we want to open our file. As we want to write the data sent by the NCBI in this file, that's why I have opened this file in write mode. And by writing as B, I am opening this file as B. Means I am giving this file a nickname. So instead of using this full name again and again, I will just call it B. Okay, and then we added colon, we pressed enter. So here, this line number 9, it only means we have opened this file, we have opened it in write mode, and we are calling it B. And then in this second line here, by till line number 9, we only opened the file. Now we have to save the result in this file as well. That's why we are opening this file. So here I have written the nickname of my file B. I have called the function write because I want to write the data sent by the NCBI in this file, which I am calling it as B. So here I write the name of my file B. I added the dot, means dot operator, which here, so here I call the function write. Then I added the parenthesis because write is a function. Now inside this parenthesis, I gave the input that from where I want to write the data inside my file. From where I want to write the data inside my file. So I want to write the data from here, result underscore handle. Okay, now by writing this, it means that I want everything that is written inside this variable, that everything that is stored inside this variable, I want to write it inside file B. But before that, we have to read this variable. That's why I here call the function read. So as read is a function, that's why I have added parenthesis. So by writing this, the name of my variable and calling the function read upon it, what will BioPython do? BioPython, look at my cursor, BioPython will read this variable, and after reading this variable, means after reading all the data that is present inside this variable, BioPython will write it in B. What is B? It is my file name. Now when I will click on the run button, we will see a file blastresult.xml here in my folder. Are we clear? Ready mode means in which mode we want to open the file. Do I want to open my file in read mode? Read mode means we can only read the file, we cannot write in it. Or do I want to open my file in write mode? In write mode, it means we can write inside the file. Okay, so whenever we are opening a file in Python, we have to give two inputs. We have to give the file name and we have to write the mode. Just like when we learned about reading the faster file, we have to write the file name and we have to write the format of the file. In the same way, if we want to open a file, we will give the file name and we will give the mode. Okay. Yes. So now here our program is complete. Now when I will click on the run button, all the work will get done. But I will not know when will the work will get done. So here I want a confirmation message that my program is working, that my program has done its work. So here I will print a confirmation message. Okay, so here I will print that BLAST performed successfully. Okay, so it's only a confirmation message. As we have learned in the earlier classes that Python moves from top to bottom. So what will happen when this program will get completed? Then Python will come here at the last line and it will print me BLAST performed successfully. Okay, so now click on the run button. Let's wait for 2 minutes. So let's revise it again what we have learned. Here we wrote our program. Here we are using the NCBI BLAST. So whenever you are writing this program, you have to use your internet, because here we have already read here that this module provides code to work with the WW version of the BLAST provided by the NCBI. So whenever you will write this program, you need your internet. So here we used this module. We called the function qblast because this function is going to send our query sequence to the NCBI BLAST. Here we passed 3 arguments: our program, that is BLAST P, the database which we want our query sequence to compare against. So here the database is Anna. Then we understood that the NCBI will send the result back and the result will get stored inside this variable. So that means we have to open a file. That's how you will build your logic. So here you just needed one fact to learn, that the NCBI will send the result back in this variable. Now that's how, after learning this fact that all the result will get saved inside this variable, that's how you will build your logic, that what to do next. When NCBI will send the result back in this variable, then what to do next? How you will read that result? So that means you can open a file. That's the only logic that you can build, that you can open a file. So here, by writing this line, here you learn two facts: that if this file already exists, in your folder, it means the file will only get open. But if this file do not exist, then what will happen here? Python will create this file as well, and Python will open this file as well. And here you have to give the write mode, that you have to open the file in your write mode. Then here you have to give the nickname, that you are opening this file as B. And then in the next line you will write that you have to write all the data which is inside this variable in this file. So this is what this program is about. And at the end you are writing the confirmation message that your program that you have written here, it is working successfully, it is working without errors. Now let's just wait for 2 minutes. Here Biopython will print us BLAST performed successfully, and here in the left side we will see this file name. Till then, if you have any doubts, you can share. So Deshna, just like I told you that you do not directly print your variable. You have to create a file and you have to save that data in a file. What if you want to see that result again that NCBI sent you? So you will just run your program again and again and wait for, right? You are getting my point? If you want to see the data that NCBI has sent you, what you will do, you will just run the program again and again. So that's not the logic, right? So you have to save that data. So we are saving that data in the file. Siddesh, can you please specify? Siddesh, can you please? Okay, so here Biopython has printed us blast performed successfully. Siddesh, what do you mean by, I understand what is VCF, but what do you mean by that? Okay, see here, we are going to see the file format, why I use the file format XML. Let's see here. So here we are saying the Biopython has printed me blast performed successfully. So that means my program has run successfully, there were no errors, and here we can see the result. So let's, Siddesh, now look at here why we use the format XML. Now I'm going to open this file. Let's open this file. See, so this is the file. Okay, this is the XML format. So you are seeing here all the sequences here. This is your sequence. Let's move down. This is your next sequence. Let's move down. So here you can also see that this is the accession number. This is the description. Here is again the accession number. And here this is your sequence. Again, this is the accession number. This is the description. This is the accession number. You can also read it as reference ID. It is similar as sequence ID and accession ID. It's all the same. Okay, and then here is the description and down there is the sequence. So whatever we saw in the NCBI blast here, in the NCBI blast, these were the sequences, right? These are all the sequences. So here in our file, we are seeing these sequences. So let's see, this was our first sequence with the accession number NP00100896. So let's check it out. Okay, so here this is our query ID. Okay, so this is our first accession number. This is the description, and this is the, here you can see the length as well. So BLAST has returned us all the sequences which are similar to our query sequence. Okay, so I'm not seeing here the accession number that I just read. It's NP. Okay, so this is the only first sequence. This is the first sequence we are seeing here. So as you can see here, these are the sequences, right? You can see these are the sequences, the length of the sequence. So these are all the sequences which are similar to our query sequence. So this is BLAST. This is how you find all the similar sequences. Okay, and this is the XML format. Do we have any doubts? Zililam, we will read that file. Today we are not reading this file. In the next session, we will read this file. Today we only learned how to find out the similar sequences to our query sequence. We will read this file as well. We will also learn how to read this file. Ridhi, what do you mean? Yes, Vidasri, you have to use XML format. Wait a minute. Sudarshana, I just here. See, this is the, okay, wait a minute. See, this is the XML format. We have learned about the formats, right? If we open a file, in if we add the extension .py, so this is the Python file. So this extension makes it a Python file. If I add FASTA, so this is how a FASTA file looks. In the same way, if I have added the extension .xml, so this is how a XML file looks. Okay. We are using the XML format because it is more structured. That's why we are using the XML format. So if you want to create the file, you have to write here XML format. I will also search for the other formats as well, and I will also let you know, okay, what other formats that you can use. Okay, Siddesh, do you want me to show you this code here? Okay, do not get confused about the format here. Today we have only learned how to use BLAST using BioPython. We will, in the next session, we will learn how to read this file, and then you will get that this XML format is more structured and we can easily parse through all the attributes, ID, sequence ID, description, everything, all the information about the sequence, we can easily parse through it. That's why we are using the XML format. Okay, just understand that XML format is most structured format to save our BLAST results. Abhay, I did not get your question. Which command for reading it? Yes, as I said that NR is the default. You understand the word default, right? So NR is the default and the safest choice that you can use in your code to compare your sequence against the protein database. Yes, Zilam, just like I told you, today we only learned how to perform BLAST using BioPython. We have downloaded this file successfully. In the next session, we will learn how to read this file. So Deshna, it's okay. I will share the question as well for you to perform this, okay? You can also share your screenshot. Riddhi, that's the same. This is the same info. If you are doing it through Blast in the NCBI, if you're opening the Google and if you're writing the program, it's the same thing. But we are learning BioPython, we are doing programming, right? That's why I showed you both how to do it in the NCBI and how to do it in the BioPython as well. Abhay, we will do that in next session. Don't worry, we will do that, okay? Okay, so one more thing. Okay, I thought that any one of you will ask me that question, but none of you asked me that question. Here, we pasted this sequence, right? Here, this was our FASTA file. Okay, when we already have a FASTA file, and we also talked about it in earlier lectures that we have a large sequence. We have sequence of 80,000 base pairs, more than 90,000 base pairs. So we have that one lesson about it, right? So, but today I pasted my sequence here. I did not use the file as I have already the file. My file is insulinprotein.fasta, but I pasted here. So let me show you how you can use your file as well. What if your file contains the sequence more than 99,000 base pairs? Okay, so you can't just copy and paste it here in your code. That will be a very long code and that will not be a practical thing to do. So how to use your file to use the BLAST? So here what we will do, we have already learned how to use sequence IO module. So here I am importing from bio import sequence IO. So I am importing the sequence IO module from the BioPython. Now what I will do, I will just make a variable name record, and here I will use the sequence IO module. We have already learned that we call the read function and we give the input of our file name. So here the name of my file is insulin protein.fasta. Then we write the file format, that is FASTA. Now, instead of pasting this whole sequence, what I will do, I will here simply write, we have already learned how to read this file. We use the attributes ID, description, sequence, right? So here in this sequence argument, inside this I will write record dot sequence. So this means the sequence which is inside the record variable. So I have already stored this code inside my record variable. So I am reading this file, Biopython will read this file and it will read the sequence from this file. Okay, so simply instead of pasting the whole sequence, I simply used my variable and I wrote dot sequence. Are we clear with this? Instead of pasting, if we have a very short sequence, then we can paste the sequence here. But if we have a very long sequence, then we will write our code like this. Okay. Okay, that's what I am talking about. Do not memorize how to write a code. Build your logic how you will write the code. Okay, it's not very difficult. You might be seeing that you have to learn many things. It's only the syntax that you have to learn. I have told you earlier that if you will not learn how to write ABCD, how you will make the sentence, it's the same way. Here ABCD is the syntax. You just need to learn the syntax and the logic you have to build it by yourself. Just like here I told you, what if you have a very long sequence? So you can't just paste the sequence here. You can use your file as well. Okay, and that's how today we use the sequence IO module, we use the sequence NCBI WWW module. We use these modules together in our program. And today we learned one more thing as well, how to open a file. Okay, so in the next class we will learn how to read this file. It will overwrite it. Siddesh, it will overwrite it. Okay, if you see here, now my file already exists and I'm giving the input of my file here and I am opening in the write mode. So what will happen if I run my program? It will overwrite. Okay, it will get overwritten. So all the data, all the previous data will get erased and the new data will get overwritten. Okay, these are in the Python foundations. So yes, we did not learn Python, everything about the Python foundations. We only learned what was necessary to learn in the Biopython. So I think we will have one Python course as well where we can learn the complete Python foundations. Okay. So any other doubt regarding our today's topic?